Welcome to Educom. This is Imam with you, and today we'll learn about Chapter 10 Cell Cycle and Cell Division from Class 11 NCERT Biology Test. Going into the class, are you aware that all organisms, even the largest, start their life from a single cell? We have learned in earlier class, that is cell unit of life, where we see means all organisms, they are composed of cells. Some are unicellular and some are multicellular, like us, like humans, animals, plants. Okay, there is other lot of other microorganisms which are made up of more than one cell. So you may wonder how a single cell goes on to form such a large organism. You might have seen elephant. It also starts from the single cell. So how exactly this the organism goes to form such a large structures? Hence, here growth and reproduction are characteristics of cell, indeed of all living organisms. So growth and reproduction, they are important characteristic of cell. Indeed means in all the living organisms, all cells reproduce by dividing into two, okay, with each parental cell giving rise to two daughter cells each time they divide. So what does it mean? Every cell it reproduces by dividing into two. So here the parental cell, that is the parent, means from where the new cells are forming, the parental cell giving rise to two daughter cells from its single cell each time they divide. These newly formed daughter cells can themselves go again, they will grow and become old and divide again. It is the same process and it is a continuous process. So the newly formed daughter cells can themselves grow and divide, giving rise to new cell population that is formed by the growth and division of a single parental cell and its progeny. So from where the new cell population, that is a lot of young population. For example, from male and female, okay, there is a progeny. They grow and they become younger and they'll produce. So reproduction is continuous along with the growth of the organism or cell. So in other words, such cycles of growth and division allow a single cell to form a structure consisting of millions of cells. Okay, you understand now? So this cycle, one cell to two cells, then 2 to 4, 4 to 8, this is a continuous process. Okay, this cycle of growth and division will allow us to see how the single cell is going to form a large animal, or which consists of billions of cells. So here important things to consider is growth and reproduction are characteristics of cell. Okay, coming to cell cycle, cell division is very important process in all living organisms. Okay, during the division of cell, DNA replication and cell growth also take. Okay, so it is not only multiplication. The cell which is the new cell which is formed, it grows, okay, during for certain amount of time, then again it will start reproduce, again it will continue, and the same progeny, the newly formed cell, they keep on continuing the doubling. So we'll be having new cells continuously. So from one cell, the new cells will be forming continuously. The new cells which become old, again the new cells are formed. This is the process. So cell division is a very important process in all living organisms. During the division of cell, DNA replication and cell growth also take place. All these processes such as cell division, DNA replication and cell growth hence have to take place in coordinated way. It's a step-by-step way to ensure correct division and formation of progeny cells containing intact genomes. Okay, so all these processes, the division, what happened first? DNA replicates, okay, it moves on to the other cell, then the cell division happen, the divided cell, the new cell, it will grow further, again it will divide and form new cells. This is a coordinated way which will ensure the correct division and formation of progeny cells containing intact genomes. The sequence of events by which a cell duplicates its genome. The sequence of events by which a cell duplicates its genome, synthesis, the other constituent of the cell, and eventually divides into two daughter cells is called it's termed as a cell cycle. What are the sequence of events? Okay, that is, one is cell duplicates its genome, okay, synthesize the other constituents like, okay, organelles which are present in a cell, okay, of the cell, and eventually divides into two daughter cells, which is termed as a cell cycle. So cell cycle means DNA replication, okay, and because from parent the DNA is shared with the young one, the new cell, and the synthesis of other constituents of the cell. What are the other constituents of the cell? Lot of membrane bound organelles which are in eukaryotes. They all form eventually dividing into two daughter cells. Okay. Although the cell growth in terms of cytoplasmic increase. Okay. Although cell growth in terms of cytoplasmic increase is a continuous process. Okay. Cell growth, that is according to in terms of cytoplasmic increase. Okay. DNA synthesis occurs only during one specific stage in the cell. Okay. DNA is not, cell growth will be there, but you'll be having lot of other things which is a continuous process, but the DNA synthesis will not happen. It occurs only during a specific stage in the cell cycle. Okay. The replicated chromosomes, that is the DNA, are then distributed to daughter nuclei by a complex series of events during cell division. Okay. So what happened first? DNA replication, then cell division, and then cell growth. Okay. The cell growth can be cytoplasmic increase also, because every time cell, will be seeing the increase in cytoplasmic content, but there will not be any new DNA form. So only DNA synthesis occurs only during one specific stage in the cell cycle. So the replicated chromosome, that is DNA, are distributed to daughter nuclei by a series of events, complex series of events during cell division. These events are themselves under genetic control. Everything, for example, we learned that about the cytoplasmic increase, even that, and the other constituents of the cell. Okay, the synthesis of other constituents of cell like organelles, different, they are all controlled by the DNA. So the events, all these events are under genetic control. So what are the phases of life cell cycle? Here, a typical eukaryotic cell is illustrated by human cells in nature. Okay, typical means you are taking that as an example. These cells divide, okay, once in approximately every 24 hours. Okay, you should remember this, human cells, okay, because average these cells divide once in approximately every 24 hours. Okay. However, this duration of cell cycle can vary from organism to organism. So this the time taking for the division of a cell is not constant. It is different from organism to organism, and also from cell type to cell type. So here you should remember 2 points here. The duration of cycle can vary from organism duration in the time taking. Okay, duration of the cell cycle can vary from organism to organism, and also cell type to cell type. Different types. For example, we have cells in bones, we have cells in blood, okay, we have cells in tissues. They all have their own independent functions. Okay, they are not all same. Okay, these two are the important lines here. So human cells, they divide once in 24 hours. At the same time, the duration of cell cycles can vary from organism to organism, and also from cell type to cell type. Okay, this is the important here. You may have a sentence like questions like, which of the following statement is correct. So here these can be useful. These lines are very useful. And also yeast, for example, can progress through cell cycle in only about 90 minutes. So for humans, how much the time is for the cell, for the division of the cell? It is approximately 24 hours. Whereas in yeast, the division, the cell cycle is lost only for about 90 minutes. The cell cycle is divided into two basic phases. Okay, how, for example, we are studying now what is the cell cycle. Then we'll see what are the different stages in the cell cycle. One is interphase, and next is M phase, that is the mitosis phase. We might have heard about this interphase. In cell unit of life, in the nucleus, we saw that there is one line. The interphase nucleus means the cell, when it is not in dividing state, contains highly extended elaborated nucleoprotein complex called chromatin. Okay, at that time there is no any chromosomes. Okay, it is present in the fiber. Okay, like thread like structure which is present in the nucleus. Fine, that word was used there. Okay, here the M phase represents the phase when the actual cell division or mitosis occurs. So here this is a pictorial representation of a cell cycle, how the new cells are formed. So here this is here, this part from here to here. Okay, there is no any division. So actual division happen in M phase. Okay, after M phase. Okay, this is the, so the M phase represents the phase when the actual cell division or mitosis occurs. So in this place, in this stage itself, the actual cell division and mitosis occur. And the interphase represents the phase between the two successive M phases. Okay, it is significant to note that in the 24 hours average duration of a cell cycle of human cell division proper lost for only about an hour. Okay, see. Then what we discussed in earlier, what I made a statement before, that approximately cell divide in every 24 hours. But in that 24 hours, the cell division proper, where exactly the cell divide itself, is about only an hour. The interphase lasts more than 95% of the cell cycle. So out of 24 hours, okay, only 1 hour, okay, is used for dividing the cell. The rest 23 hours is in the interphase state. Okay, the M phase starts with the nuclear division corresponding to the separation of daughter chromosome, which is also called as karyokinesis, and usually ends with the division of cytoplasm. So here you can expect a sequence like question. For example, what is the correct sequence? Karyokinesis, cytokinesis, like that. Then first cytokinesis, then some other, then karyokinesis. So we must remember this. The first in the M phase starts with the nuclear division. The M phase starts with the nuclear division corresponding to the separation of daughter chromosomes, that is karyokinesis. The separation of daughter chromosome is karyokinesis, and usually ends with the division of cytoplasm. The interphase through called the resting phase, okay, interphase is also called as a resting phase, is the time during which the cell prepare for division by undergoing both cell growth, also DNA replication in an orderly manner. So where exactly the division exactly occurs in M phase, but in interphase the cell will prepare itself for the division by undergoing both cell growth like cytoplasmic growth and also DNA replication in an orderly manner. Again, the interphase is divided into 3 phases. Okay, G1 phase, that is gap 1, S phase, that is synthesis phase, G2 phase, that is gap 2 phase. Coming to G1 phase, G1 phase corresponds to the interval between the mitosis and initiation of DNA replication. Okay, DNA phase, you will see the picture for you to understand properly. So here, this is the G1, This is the S phase and this is the G2 phase. This comes under interphase. This is the M phase here. This is mitosis, where the exact cell division and cytoplasmic growth, everything occurs in M phase. Okay. So G1 phase corresponds to the interval between mitosis and initiation of DNA replication. Okay. During DGN phase of the cell, during the DGN phase, the cell is metabolically active and continuously grow. Growth is continuous. Okay, this is important point. Okay, but does not replicate its DNA. In G1 phase, okay, there is no any DNA replication, but we see the cell is metabolically active and continuously grow. So in G1 phase, we saw it is that GN phase correspond to the interval between the mitosis and initiation of DNA replication. Okay, here DNA replication doesn't happen, so the cell will grow continuously. Okay, then in S phase or synthesis phase, S phase or synthesis phase marks the period during which DNA synthesis or replication takes place. Okay, in this phase, remember, S phase or synthesis phase, it is the period during which DNA synthesis or replication takes place. During this time, the amount of DNA per cell doubles. Okay, so here doubles, it is mentioned clearly. So this point is very important, quite important to remember. Okay, DNA synthesis or replication takes place in S or synthesis phase, and also during this time, the amount of the DNA per cell doubles for it. Okay, if the initial amount of the DNA is denoted as 2C, then it increases to 4C. However, there is no increase in the number of chromosomes. Okay, if the cell had diploid or 2n number of chromosomes at G1, even after S phase, the number of chromosomes remains the same, that is 2n. So what do you understand from this? Here there is no any DNA synthesis occurs, but it doubles, but it doesn't mean that chromosome size numbers are increasing. Okay, the size of the chromosomes increase, but DNA is increased, but number of chromosomes is affected. If the cell had diploid 2n number of chromosomes at G1 phase, even after S phase, the number of chromosomes remains the same as 2n. Okay, only thing is here there is no any kind of doubling number of there is no any kind of replication forming new chromosome. Okay, in animal cells during the S phase, DNA replication in animal cells, okay, it mentioned here in animal cells, during the S phase, DNA replication begins in the nucleus and the centriole duplicates in the cytoplasm. Okay, so centrioles, we learned that they help in cell division. Okay, that's in earlier chapter. So in animal cells during S phase, DNA replication begin in the nucleus, but centriole duplicates in the cytoplasm. Okay, during the G2 phase, proteins are synthesized in preparation for mitosis while cell growth continues. Still, the growth of cell is continuous in the G2 phase. So till now what we finished, G1 phase we have finished. Gn phase is the interval between the mitosis and initiation of DNA replication. There is no any kind of DNA replication in G1 phase, where the cell remains metabolically active but continuously grow, but will not replicate its DNA. Whereas in synthesis phase, the DNA synthesis or replication takes place. During this time, the amount of DNA per cell doubles. Okay, if the initial amount of the DNA is denoted 2C, then it increases to 4C, but there is no increase in number of the chromosome. Coming to G2 phase, proteins are synthesized in preparation for mitosis while cell growth continues. So in G1 phase, proteins are synthesized in a cell to prepare the cell for mitosis, and the cell growth continues. Some cells in the adult animals do not appear to exhibit division. Okay, example, take pod cells. Okay, some cells in adult animals, like in any animals, so they do not appear to be exhibit division, so there is no exactly division of the cell. And many other cell dividing only occasionally when required, as for example, if there is any damaged cell, okay, in that organ also, at that time they divide as needed to replace the lost or damaged cells because of injury or cell death. These cells that do not divide further exit G1 phase. Okay, G1 phase. Okay, you remember? So such cells which will not divide, they will exist at G1 phase itself to enter an inactive state called quiescent stage, G0, okay, of the cell cycle. So here hot cell should be, this line is very important, because hot cells, they will divide occasionally when there is a damage, injury or cell death in that particular organ. Okay, cells in this stage, that is G2 stage, in this stage remains metabolically active but no longer proliferate, that is a GO, this, okay, GO cycle, GO phase, that is a quiescent stage. Cells in this stage remain metabolically active but no longer proliferate, means multiplication, okay, proliferate unless called on to do so, depending on the requirement of the organism. So if there is a damaged cell, that is what this sentence, makes us to understand. In animals, mitotic cell division is only seen in the diploid somatic cells. Okay, in animals, mitotic cell division is only seen in the diploid somatic cell. However, there are few exceptions to this, where haploid cells divide by mitosis. Example, male honeybees. So here we should remember these 2 points. In animals, mitotic cell division is only seen in the diploid somatic cells, whereas you can take certain exceptions, like in male honeybees, where the haploid cells divide by mitosis. Okay, against this, the plant can show mitotic division in both haploid and diploid cells. Okay, from your recollection of examples of alternation of generations plants, that we'll discuss in chapter 3. Okay, so now with this we conclude this small session, and after finishing this video, after listening to the video, kindly go through your NCERT book, okay, and read it once. The more you revise the book, the more you gain the subject. Okay, and thank you, and please subscribe to my channel EduCon.